This is a production of Cornell University.
Well, thank you, Kathy. That was a most wonderful introduction. I feel kind of embarrassed. I didn't think I have done those things that you mentioned when you. I am, as Kathy said, I am an expert on taxonomy. I'm a systematist, and I work on naming and describing and deciding the names of organisms, and also understanding their phylogenies. That's what I've been doing since the start of my career with Professor Korf, and of course I have moved and evolved in the field as it has evolved itself. I'm terribly excited to be back at Cornell, and I know some of the faces here were my professors or my colleagues then, and it's absolutely wonderful to be back at the herbarium where there's so much to do. Just entering that herbarium is entering into one of the most precious places in the world because of all the wonderful wealth of fungi that are there, not only from the region, but also in the Discomycetes. So my work I work mainly with fungal diversity of tropical fungi, and I will not say only Discomycetes because some of my students back in Venezuela were interested in other groups that were not Discomycetes. So I had to broaden my interest and work with other groups as well. I also work with ecology, life history, and fungal communities. I try to understand the biology of these fungi. What are they doing? Why are they in that environment? For this talk, I want to wrap up a bit and place myself where I come from in my lineage of Cornell mycologists, because as I stand, as they say, in the shoulder of giants, and it's true, I totally do. And most of them, in my case, has been Cornell mycologists. And one of the goals of this talk is also to excite you and to make you very interested in CUP herbarium and in the Cornell herbarium, so that you will deposit your specimens there. And you'll understand after this talk why is that so important. So just to start with fungal diversity, this is a slide you must all know very well. There's all this saying about how many fungi are there. Are there 1.5 million? Are there 5 million fungi? Every time somebody goes and samples, does environmental sampling, all of a sudden from 1 centimeter of soil, there comes 100,000 sequences. So nobody really knows. But if there are that amount of fungi, which probably there are, we know very little about them. We know less than 5% probably of what's out there. And we understand, even as time goes by, we understand less and less about these organisms, because now it's all about sequencing, and people have forgotten how to describe or how to even look at their organisms in the field, less understand their biology. So that's what I do. I try to understand the whole picture. I go out to the field, I collect these fungi. These are all cup fungi. As you can see there, some of them maybe you will recognize as, for example, this is a beautiful anthracobia, which we will be talking about. And these are Pezizas, and this is Bisporella. Some of these fungi operculate and some are inoperculate. And I work on both groups. So the first group I'll mention is the group of operculate fungi, the Pezizomycetes. And this is a real picture. It's not fake. Can you imagine a frog just sitting there and the person being there at the time where that happened? I always dreamed it would happen to me, but it didn't. This is a photo that somebody sent to me from Borneo. It's a collaborator that's working with a group of us in a beautiful genus that I will mention in a little while, that's called Kukieina, the name over here. So this comes from Borneo, as I mentioned. These operculate discomycetes have the characteristics that their ascus opens by a lid. It just pops up and pops open, and the spores are dispersed in that way. That lid can be on the top or can be a little on the side. The vast majority of these fungi, the vast diversity, the highest diversity, is in temperate regions. But in the tropics particularly, and in the tropics throughout the world, the genus Kukieina, which is of the group that has a lateral lid, is very, very abundant. So for years, and still up to date, we have been studying the genus Kukieina with a group of people. And since 2002, we thought before 2002, we thought there were only 4 species, which were all that had been described. So there was a very hairy one that we called one name, a little one that was less hairy, like this one, the Kukieina speciosa group, which we noticed had a lot of difference in colors. But when we went to the microscope, it looked the same. So we called it Kukieina speciosa. And then a third one, which was a little smaller. And then another one, which I'll show you the pictures. There were 4 species in the world. We almost didn't care to look very much at the microscopy because we thought they were those 4, and that's it throughout the world. In 2006, we started working with colleagues from the Farlow Herbarium at Harvard, Don Feaster. We started working with molecular on these groups of fungi, and I was in Venezuela then, looking at all these different color variations on this Kukieina speciosa. And we started noticing things. So we started understanding a bit more and going back to the morphology. So since 2006, only 3 species have been described. And it's not because we are sloppy, it's just because we still do not understand what's going on with that group. This is just the Kukieina speciosa complex, and I'll show it ranges from all colors, but that's the only thing that we see, colors and geographical variation. There's no morphological, as we start the morphology under the microscope, there's nothing that that tells us there's any differences. But when we have done sequences of this, we can see that the speciosa that's up here has different clades, and these clades really relate with colors, and which I have not marked there, but this is, for example, is the corals. These ones are the more yellowish. These ones are the whitish and the brown one. So there are some differences. So people at the beginning, when they started describing all those names they had for each color, and I'm talking about even the 1700s and the 1800s, they were right. There are all these names which we're going to probably need to go back to and rescue. But we have been going back to Kukieina, looking them under the microscope, and we have been noticing a few things that we had not seen before. We have been noticing some difference in the hair, some differences in the structures, and some differences also in the growth, and who knows how many other differences there are in their physiology, which we have not even looked at. So Trichaleurina is a very homogeneous clade throughout the world, and this is the clade where Kukieina incititia is, which doesn't mean much to you at this point. I just want to show you, this is an edible fungus. It's edible. It's not a big fungus. It's about 2 inches height and 1 inch in diameter or smaller, but it's the abundance that and because of its colors, when you're in the Amazon, for example, in low tropical forests, it occurs so abundantly that you can even pick them up like if they were seeds or flowers. So they are edible, and in Mexico, they're even sold in the market. I didn't believe this the first time that's in an Indian community in Venezuela, somebody told me, and I said, oh no, you must be wrong. And they didn't tell me anything else, so I totally was in disbelief until I heard, of course, that yes, they were, and Actually, I've tried them and they're pretty, they don't taste like anything, but they have, but they have protein. So this is a beautiful drawing by Madame Legal, a expert on the discomycetes, where the intricacy of this paraphysis is shown, and also the striation of the spores, which is an important character. This fungus is very particular. This is only the Coccina speciosa complex, so that you can see the color variation again from white to brown, from all these incredible colors. So this is Trichaleurina. You can see that the long hairs, it's very easy to identify, and the clade is very homogeneous throughout the world. There's just a specimen from China, which has smooth spores, and it's very related to it. And the last clade that we have is a Coccina incititia clade, which it comes together with the other species that we know. And this one occurs only in Asia. There are some cautions when studying this fungus. If you ever go to the tropics, and there are several programs where Cornell takes students to the tropics, so you'll be able, if you're there, you'll be able to find one. If you're in the lowland tropics, you will find Coccina trichaleurina and speciosa, the two that I've shown mostly. But there's a third one called Coccina Venezuela, which can also, that occurs in highlands, in cloud forests. And that, I watch, so you may be able to have sometimes in your life to look at this fungus under the microscope. And I want to make sure that you remember that color is important, that geographical region is important, and also that in this particular family, there's a phenomenon which doesn't occur in other groups, which is that ascospores mature simultaneously. So in each apothecium, in each fruit body, all the spores will be in the same state of development. All the acai will be in the same state of development. Professor Korf always was very excited with this fact, because it doesn't occur in other groups of fungi. So it's probably something very interesting to study evolutionary by some of you. But this has led to problems in the descriptions. Some people have described new species based on immature spores, because that this is not something we see in general when we look at an a fruit body. Spores are in all different stages of development. And about what do we know about the biology? Well, we know very little. At a certain point, I was placing colored pins besides each apothecia. I did this for years, but this was in the Amazon, and then there was a huge rainfall and everything got washed away. So then I did it with another species that occurred at my university in Venezuela. Again, the same thing happened. These things grow on little twigs and little seeds. So, you know, you can imagine having a pin next to it and trying to watch its development throughout time is not very, very easy. So after maybe 1 month, they are resistant, they are long-lived. Nothing had happened. But we do know there's an imperfect state called Leotiomycetes that was described. And as you know now, there's one fungus, one name. So now, of course, this is called, would be called the asexual state of Coccina, in this case, speciosa. And this one fungus, one name is a big decision that was that happened in Melbourne, in the code, in the meeting there in Melbourne, already in 2014. So it's been quite a few years that this new rule has been there in the Code of Nomenclature. I meant to bring the new code to show it to you, but I have it in the office if you'd like to look at it. So the second group of fungi that I work with are the Leotiomycetes, and these are the inoperculate discomycetes. So this, in all of this group, there's supposed to be a very homogeneous kind of spore discharge, which is by a pore. By a pore, in the apex of the ascus. So through that little tiny pore, which frequently stains blue under reagents that have iodine. So this pore is supposed to have some starch in it. It has been used in taxonomy to divide these two groups, and it holds phylogenetically. This is the highest diversity of cup fungi are in this group, and it's much more diverse than the Leotiomycetes worldwide. The thing with these groups is that in general, they're very, very tiny. Some of these are beautiful and have been really used throughout history. For example, Chlorocyboria, which stains wood green, has been used in making furniture. And when one sees those beautiful pieces of old furniture where there's all these little flowers, for example, made in wood, whatever is green has been made from wood where the mycelium of this fungus is included. And these are other Leotia that you know, and Bisporella, and one of the Hylocyphaceae. So one of the fungi I've been working in the last few years, the last 2 years, it was published, I think, last year, is this fungus, which is kind of bizarre. I had it on my post on FaceTime, and somebody asked me, "What's that horrendous fungus that you have there on your post?" But actually, it's very interesting, and the interest of this fungi is that I looked at it for a lot of years, and I just started looking at it, and then I left it there. I was continuing with other things, and then I came back to it, and there was something wrong. And its placement does not prove anything. We did ITS sequence and LSU, and also the elongation factor of this in this tree, and studied other genes as well. And it just comes somewhere there in the Leotiomycetes. So there's no placement. And why is there no good placement for this fungus? Because there are very few of these fungi that have been sequenced. So really, every time that we work with one of the Leotiomycetes, we find that it comes to something. It doesn't give us much information. Phylogeny doesn't really give us much information, because in the case of Bulgarella pulla, there is an ascus. It has brown ascospores, and it has weird paraphyses. And I could tell you much more about this fungus, but I will not. I will just tell you that the way the ascus breaks is very particular. It doesn't break through a pore. It just breaks like another totally different and unrelated group of fungus, which is by the total break of the rupture of the spore wall. So at the end, we end up just with two walls, and the apex of the asci disintegrates, and that's how spores are released. There is here, you can see, and little by little, this took years to understand, many months at least, of understanding how all this process started and continued, and how the wall broke until only the two walls were there. And this has not been described yet. Lots of people had looked at Bulgarella, and this only happened because I looked and looked and looked for a long time under the microscope, even to the point where I thought, why am I looking so much? Until I understood this happened, and it was published like an important thing, because this had not been recorded yet. So it is important to look. Another issue I've been very interested in since my PhD work, and that was in 1990 under Professor Korf, have been the connections between the sexual and asexual states, which before each of them, as I've already mentioned, have their own name. So I studied this little fungus due to the persistence of Professor Korf, because this fungus is 1/4 millimeter in diameter, very difficult to find in nature. And it took a lot of going throughout the world and a lot of time in the field in order to find a few of the species. So we did a monograph on this fungus, and also looked at its anamorph, as its asexual form, which was called then Pseudospiropis. Now it's called Strosmeria. This is a very uncommon fungus in the sense that they're usually not brown dematerious anamorphs connected to Leotiomycetes. They're very few. And also there are other characters in this fungus. So in this sense, I have been working at that time. How did we do the connections? You must wonder. There was no molecular involved. So there was no way to know. We did cultures very carefully, cultures of the ascospores of the sexual body, and the single ascospores, and weighted. Is it homothallic or heterothallic? Is there something there? So after months, when we had totally forgotten that the cultures were there, all of a sudden, Professor Korf was shouting, "It's growing, it's growing!" And there was the Pseudospiropis growing there. So again, connecting these anamorph and teleomorphs, there is this fungus called Trichaleurina javanica. And last year, I went to Mozambique in a very, very bad time of the year for a collecting trip I had been invited to. And this is, you know, a national park, Gorongosa National Park. And one has to collect with a guard because of the elephants and other animals. So nobody's permitted but to be on the jeep. So it's very exciting because you see an elephant over there, and you think there's a tiger going to come to attack you, but nothing of that happened. We were scraping the floor, scraping everything because there were so few fungi. We were really depressed. And why did we come in this time of the year? Until we started finding things that were totally unusual, that we started finding this hairy thing. We didn't know at all what it was. I sent it to some colleagues, experts in anamorphs. Nobody knew what it was. So we grew it in culture. It grew beautifully, lots of hairs coming from those Petri dishes. And we were able to sequence it, and it sequenced, as one of the few times it happens, identical with 100% similarity to Trichaleurina javanica, which was only known by its fruit body. And just a few years before, there had been somebody working on the sexual state and had been able to obtain the fungus in culture, the anamorph in culture, but had never seen it in nature, neither knew anything about where it occurs. So that's pretty exciting when that things happen. This is, I am not going to mention other examples, and there are many, of this anamorph teleomorph connection, which now we are lucky to be able to do using molecular, if there has ever been molecular, its DNA has been studied. This usually the case is not that. Usually it lasts to nothing. So then we describe it as, again, the old method. So the third thing I've been working with are fungal communities. And also last year, there was a big fire in the Smoky Mountains. And it was a fire that started in 2016. We started studying it in 2017. It started in November, and it was contained by mid-December. People died, structures were lost. It was really, the town of Kattenberg was, when we started going there in March of 2017, it was very sad. Of course, you've all seen the recent fires on the West Coast, and that's not unusual for the West Coast, but it is unusual for the East Coast. Well, it's sad to say that probably with the weather changes, etc., and I don't want to use the word climate change, but it may happen that we get abundance of water or more fires also. So when we started going there, this is what we saw. There was nothing, but that's not true. There was really, when looking carefully at the soil, fungi in March were already growing there. And fungi, specifically cup fungi, and some of the agarics were already growing there. So we went for once a month during a whole year. And we understood there was a fungal succession present there. Many fungi repeated themselves. And some of these fungi were like, where are they coming from? All of a sudden, they were particularly to a fungal community that's already known to occur. So it's some of these fungi are already known from the Western United States. And all of a sudden, did they come from the west to the east? All of a sudden, through the air? Are they all around and then just land? So I'm going to talk a little about the hypothesis that exist about how are these communities, how do these communities occur? So some of the questions were, these fungi, are called pyrophilous fungi, fungi that grow after fire. They're friends of fire. And this is Geopixis carbonaria on the top. This is morel, and I have some in my lab if some of you want to see. I'm not giving out anyone because I'm going to eat them myself, but I've been waiting for a special occasion. So I may make a dish at some of the departmental parties and we can all eat it. So what is a fungal community like post-fire, and what explains that distribution? There's, as I said, we found like 50 species specific to burn sites, and that's a succession that was evident, especially on year number 1. On year number 2, there's forests growing again, and there are many other things growing. But we did find new records, and some of them with totally unresolved taxonomy still being worked at. So this is the group of ascomycetes that we found. Specifically, I'm working right now with Anthracobia, which is bringing, again, some of the same things that happened in Cochiaina are being repeated with Anthracobia. Morchella was the first report for, in the east, this species of Morchella exuberans. And in the Pezizomycota, there were new ecology things that started appearing in spring that were known only from other seasons, as the same as Mycena, and Foliota highlandensis, which was everywhere, was in a very much smaller form. I do not study agarics, but there's a huge group of colleagues from the University of Tennessee working on this. So these are the most frequently the Foliota, Geopixis carbonaria, everywhere. And these fungi, which are growing clusters, and they're difficult to get out of soil because a whole clump of soil comes with it. So one can start understanding why are they there. This is the first report of that Morchella, which was already published, and I have mentioned. And the interesting thing with Anthracobia is that there are also different colors of Anthracobia, different morphologies in Anthracobia. And on doing the Anthracobia phylogeny and on working on several genes of Anthracobia, this is only ITS, we can see that there are different clades. And here in a very rough manner, just to start with, one can see that probably there are these species present, but there are other clades in between which we know nothing about. And this is probably not Anthracobia in Ecuador, but another genus. And there are other things involved there which have been very mixed up. So at this point, we're trying to solve its, and we have gone back to trying to understand the morphology of these groups. And also we're finding lots of cryptic species in there. So there's a lot that we're working on and we don't understand yet, only about this genus in particular. So I think you'll be interested in these 3 hypotheses that I mentioned already of what explains the distribution. And I'll just mention them very briefly. The first one is the spore dispersal hypothesis, which says that these spores come from elsewhere, and finding an environment totally with nothing, with no competition at all, they start growing. But this hypothesis has not been very, if true, genetic diversity should be low in the population, and spores should germinate easily, and none of these things happen. The second hypothesis says there is a spore or sclerotium bank that doesn't get eliminated when fire passes through. I'm sorry. And then they're dormant until fire comes and they start growing. And the third hypothesis is the one that we are working on, some of my colleagues and me, which is called the body snatcher hypothesis. And it says that fungi are present there inside plants. So these plants, we have sequenced, my colleagues and myself, we have been working in sequencing mosses and sequencing lichens from the Smoky Mountains and from other parts of the world. And the preliminary results prove that they are present in mosses, and that these same fungi are present in mosses, not only on the Smokies, but elsewhere. So now there's environmental sequencing going on. There have been, and I do not have the results with me because they have not been published, and they're still being worked by colleagues in the University of Tennessee and Illinois. And it's really their work. So, but they are there and they're present. So this will probably be the most common hypothesis, the most real hypothesis, that they survive. How do they survive? Well, nobody's sure yet, but it can be related to the amount of water that mosses have, or that rhizomes maybe. Fire sometimes passes very quickly, and the rhizomes are there, and who knows, there may be a combination of methods. There might be also some structures that persist on the soil, though it's more uncommon, because here in the eastern coast, that particular region had not had a fire in hundreds of years, for example. So so that's a question for you. And the last part of I'm switching now completely to talk about herbarium. And why do I talk about herbarium? Oh, this beautiful fungi that you've seen there end somewhere. And they're important to be there. It's important that they end somewhere, and that they end as a collection in a herbarium. So this is photographs that taken from the CUP herbarium, which is a Cornell University plant pathology herbarium that used to be housed in this building in the 3rd floor. And it's not too far away. Some of you have been there, and it has a wonderful space of its own now in Game Farm Road. So there are all sorts of different kinds of specimens, starting by the Passepartout, which are just for classes, for showing diseases, etc. To students. They're mounted in a way with cotton, etc., that are very easy to pass around and to show them, and you're welcome to use them. It doesn't have only fungi, it has bacteria, it has viruses, it has all sorts of plant diseases that they're very, very nicely preserved in this Passepartout. There are thousands of Passepartouts. So the most of the collections, starting with Whetzel, are they are in these huge boxes, and these are some of the Atkinson's slides. So there are about 500,000 specimens at CUP Herbarium, fungi from all over the world. And a new project where the CUP Herbarium has been involved since, well, I don't know, 10 years ago. It has been the digitization of herbaria. So the mycoportal is a project that I was involved with, and CUP Herbarium has been involved with for many years, doing macrofungi and microfungi. So now all of these specimens are digitized, and 85 institutions from all of the United States have participated, and from other parts of the world also have participated. CUP had, and Kathy Hodge had a very good idea in digitizing not the specimens themselves, but all the card catalog that was there thanks to Whetzel and Korf. And so the whole herbarium was able to be digitized, in the sense that people all over the world, because this is of open access, can see what's there and can ask for specimens that we have. So we keep getting requests for fungi that we have and that people only learn of because of the mycoportal. In the mycoportal, there are almost 4 million records from all of those institutions. And I just did a little sample that took me 15 minutes because there are several tools in the microportal where I just said, I want to know what's the Latin American fungal diversity. And it just popped up all these numbers. Of course, this only is including specimens that are in herbaria and the specimens that have been digitized, usually georeferenced, of course, because we know they're from Latin America. So, and imaged sometimes. So all of these countries, the number of records and the number of species. Besides that, the microportal can permit making heat maps. So, and that's also done in minutes. So it's amazing when one tries this and what all the information that one can retrieve from this portal. And I did just because, of course, I'm from Venezuela. I was interested in knowing the heat map from Venezuela, my own country. Well, this is a little further down than Venezuela, but it includes, because Venezuela usually should end here, so there's must be some little problem there with the microportal. I shouldn't tell you that, but it's true. But here, the problem has been solved. So this is Venezuela, and each of these, that's another kind of map that one can do in seconds also, where each dot represents a collecting spot. Just clicking on the dot, you'll find the species, the collector, and the locality. So it's really a wonderful tool to use. With this tool, one can learn also how fungi are moving throughout the world. Is climate change affecting some diseases, and how some diseases are moving from one place to the other? When did a disease start and where? So there's a lot of information that can be retrieved from the literature and from the microportal. So voucher specimens are a vital source of information. Nowadays, we can, of course, obtain DNA from specimens that are hundreds of years old. And new techniques, if that did not happen when I was a graduate student, when I was sitting down, listening in this same room, like you guys, some of you guys are there. We didn't have those tools. So in, I won't say how many years, this has changed. Can you imagine in 50 years, 30 years, 100 years, what information will we be able to retrieve from those samples? So that's why we care for these samples. That's why it's important for all of you to deposit your samples there, the organisms you're working with. Sometimes we get some organisms deposited there, but very seldom. So please count on the herbarium. There are many questions that can be answered, and you're welcome to come. We have great facility, we have space, we have a dissecting scope, so it's there for you. And we welcome anything that you be interested for your research. Oh, this was supposed to be before this slide that but I will talk about a little, just very quickly, about the lineage where I come from. And as I said, I stand in the shoulder of giants. And this is the Cornell lineage. You know, all these people, which I will not name, with their students. So Dudley, Atkinson, Whetzel, Fitzpatrick, Korf, and that's my name over there. We all come, and we call each other brothers. So I call, for example, Don Pfister, my mycological brother. And I don't think I'm going to have time to talk in detail about some of these people, but Dudley was the first one, and I just want to say that there was like a tradition, like Dudley was the professor of Atkinson and Durand, and he has this beautiful fungus named after him. And Cornell, first president, purchased Cornell first herbarium in 1870, 5 years after Cornell started. Why was that important? Because having collections was a pride of an institution. Having collections and having museums was, as it still is, important. So, and people knew it. So Atkinson was, as we know, this wonderful and incredible person who was a student of what had Durand, Fitzpatrick, and Whetzel as his students, and was a wonderful photographer and recorded and did this great book on mushrooms. Whetzel, how can we not know Whetzel? We're standing in the room called after him. He was the father of Cornell plant pathology. He established the first department in plant pathology in the United States, and the plant pathology herbarium. So he was the professor of Fitzpatrick and Durand and worked with sclerotinia. Well, he traveled extensively throughout the world and had collaborators that you can see over there. I just want to mention Durand because we have in our herbarium the Durand herbarium. All Discomycetes he studied here, but then went elsewhere. But then he donated his herbarium to CUP. Fitzpatrick was Professor Korf's professor, and he was responsible for the herbarium and also taught a lot of mycology courses, which then, of course, Professor Korf, which died 3 years ago, and many of you may have known him, he continued the work of Fitzpatrick, wonderful work in the herbarium. The herbarium grew throughout the years with Whetzel and him until the more or less 400,000, 500,000 that it has today. He did an incredible amount of publications, as you can see there, was a member of the nomenclatural committee for fungi. And when he retired, I took his place, but in that committee. And he was also a talented actor. He was very good in acting, as can be seen here in his photo when he was a student at Cornell. And this is not how he dressed to come to classes, don't think. He was here impersonating Elias Fries, one of the very important mycologists. And he did that and was asked several times to impersonate him in several international congresses. And he loved it, and he really was very good at it. This is a photograph. Richard P. Korf was a president of the Mycological Society of America. And this is a photo that Kathy took of him on his 80th birthday, where there's Professor Korf, and there's some of you there, and I can see one person over here, which is Professor Norbier standing at the door. So you see, the tradition continues. Well, I've come now to the acknowledgement part, and I want to, I will not go through the whole acknowledgements, but I want especially to thank Kathy Hodge, Professor Hodge, for inviting me to give this talk, for her many insights into this talk, and also for being the major person that brought me back to Cornell to be a curator at the plant pathology herbarium. I'm delighted to be here, and we have a lot of projects that we're already starting, and we hope many of you will get excited and also start working with us. And I want to thank all of these people that you see there, which have collaborated in different projects. Donald Pfister from Harvard, Andy Miller from the University of Illinois, besides all the other colleagues, Karen Hughes, also from the University of Tennessee. They have all been relevant people. Meredith Blackwell in this presentation in different ways. So thank you to all. And I end with this picture of Venezuela, one of the tepuis, which we cannot go at the moment, but we hope to be able to go back in the near not so far future.
Any questions for Teresa?
So what is the ecological significance of colors with the cup fungi?
Well, there have been many hypotheses, and some of them are dispersion. There are slugs, and there are lots of insects that feed from the hymenium of this fungi. So there may be it may be a color may be there to attract them, also, it's been said a lot that they're secondary metabolites, and there are really toxins that accumulate in that the fungus doesn't have a better way to get rid of, and that's a theory that I have read. I myself have not studied it, but there are all these sort of relationships. There are also mammals that feed from fungi, and the color may have some attraction towards them, but mostly we see worms and arthropods and little insects usually feeding on them. But you know, there are not too many studies made on them, and on the Coccina speciosa specifically. Why are they growing just one next to each other sometimes and having totally different colors? Now we know that they may be different species, but what is the color significance there? You know, you can find white and brown and oranges and all of them. We thought it could be related to light at some point, but it's not. So we really and truly, to answer your question, we have not much idea. And it may differ, of course, with the group that we're talking about.
I had a question about getting your insights on the one fungus nomenclature. And I know a couple of the agriculturally significant species, there have been some heated debates on what the name will be. Are there some guiding principles on choosing what name is chosen?
Yes, and there is a whole code that I have in the room next door, and I have all the codes of nomenclature. These debates are very, very heated and very, very long, as you're saying. Only to take the one name, one fungus, one name took 30 years. So you can see there were all sorts of debates in the congresses and the panels and open panels and even votes until finally it came to that. There's still now, so now at a certain point, it was decided that the sexual stage would be the prevailing stage, but not anymore. So the following Congress, so lots of people said, why is the sexual stage going to be more important than the asexual stage, you know? So let's go by priority. Whichever name was described, was erected first, that should be the name. But what about all the fungi that plant pathologists know by one name, or that people in medical mycology know by a particular name, or that have economical importance, of economical importance? So there have been committees created to avoid these really horrible fights. There have been committees created for each of the different groups, and these subcommittees intensely debate and decide together with lots of experts which name should prevail. So these are called now conserved names. So these lists are of conserved names, just to make them different to sanctioned names, which is another group of names that were erected by Fries and Persoon, and they have their own status. So, but still, those names have the ability to be discussed. So if somebody, and those are proposals, so somebody is not happy with the name that all of a sudden the organism has, the person presents a proposal and then bases it, and then we discuss it at the committee where I am in, and that takes forever. And we vote one time, and then we vote a second time, and we vote a third time, and then a year after we come to it, and then finally at the congresses now, not the botanic congresses, but now a rule has changed also, and we are able, my colleagues are able to change the rules in their own international mycological congresses, which are every 3 years. And of course, then they have to be approved in the Botany Congress. So we are still, you know, little by little getting there. If you want to ask me about DNA as type specimens, no, it's not yet possible. You cannot erect steel. That was debated a lot in the last Congress, and some of you were there, and it didn't pass. So, and there are a lot of, I won't get into that, but I will be very happy to talk to any of you about why it was not approved. It will be eventually approved, but not yet.
Teresa, you showed many trees indicating relationship of one taxon to the next. Seems like a fantastic amount of fungal sequencing has been done. Can you comment on just to what extent sequencing was done of these thousands of different fungi?
Well, now everybody's sequencing. And we, not only the mycological community or the experts, also amateurs are sequencing all the time. So, so that's why we're having more and more sequences. And amateurs have had a very important tool, have been a very important group in sequencing, because nowadays you can send a sample somewhere, and for $30 or sometimes less, you obtain a sequence. So of course, of particular genes, but amateurs have become very good at this, and their associations. So that's why you see that amount of sequence. And we've been already sequencing since 30 years ago. So some of these trees that you saw are one gene, some are 5 genes, and now there's a tendency and a huge project to sequence, at least try to sequence the whole genomes, which amazingly doesn't cost that much. You know, in money value, it costs about $300 to sequence a whole genome. And so that's that. Of course, it's not little money for any of us, but if for a project, for example, that makes sense. So what you see is true. There's a whole lot of sequencing that has been done, but there's so many fungi. There's still so much more to do.
So yeah, we have a lot more work to do. So, but we'll let you guys go while we do it. Join me in thanking Teresa.
Thank you. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.